Welcome back, students. In the previous class, we discussed about the process of transcription. That means how the mRNA is formed from the DNA. That means how before the formation of the protein, the copy which is essential for the formation of protein, that is mRNA. So how it is formed from a DNA segment. And you have to remember one point, one gene is responsible for expression of one protein. So assume total 30,000 genes are there, approximately 30,000 known genes, but unknown genes are so many. So at least how many proteins are there in our body? How many different types of proteins are there? Imagine once. So these discussions, detailed discussions yesterday we completed. How the transcription process, how different enzymes present in eukaryotes and prokaryotes, and the location of transcription in prokaryotes and eukaryotes, and the process of transcription, how the mRNA is formed in the nucleus and how it is modified into the fully mature form before it is leaving the nucleus. Then only the mRNA is protective against the enzymes which are present in the cytoplasm. So these are all the discussions we completed yesterday. Today, before going into the translation topic, so up to here, this is transcription, and from here to here, this is translation. So before going into the translation topic, we will discuss today about the basic structure of protein. We have to know the biomolecule, how the amino acids, which are the polymers of the protein, how the protein is formed. So we will see the basic features of the protein structure. Then we will see how the protein is manufactured from the mRNA. So that process is translation. We will see the translation later. First of all, we have to know the basic structural details of a protein. Actually, these are present in the biomolecules chapter. Biomolecules, that is 11th standard topic. So actually we didn't complete that one. So if you don't know about the simple structure of this protein, we cannot understand the topic of translation. So here we will see protein biochemistry. Today's class is protein biochemistry. So what are the molecules which are present in the protein and how the protein is made up of? See here, we have to build a wall. So what we will do? These are the bricks. And in between the bricks we have cement. This is another brick. So this is cement. So here the bricks, or we can compare the bricks as amino acids. So if bricks are not there, we cannot construct a wall. So the structural and functional unit of a protein is amino acids. And the linkage between two bricks, this is first brick, this is second brick. The linkage is established by here cement. That cement here in the protein biochemistry, that is peptide bond. We will see what are the amino acids, what is the basic structure of an amino acid, and how the peptide bond is formed between two different amino acids. So the brick is an amino acid and the cement in between the two bricks is an peptide bond. So this is how we will construct a wall of a protein. So total wall is a protein. So how the protein is manufactured? Protein is manufactured with a group of amino acids: A1, A2, A3, A4, A5, A6, A7, A8, A9, A10, A11, A12, and again the linkage between here. So due to this complex bonding, a complex protein is formed. a complex protein is formed. Nothing is there in the protein structure, only amino acids. How they are folded? They are folded in one layer, they are folded in two layers, they are folded in a ball-like material. That much complexity increases from one grade to another grade. So, depend upon the complexity. There are different structures of proteins. That is primary structure. Primary structure means simple layer. Secondary structure. Secondary structure means sheet-like. This is all one protein. Tertiary structure, a small ball-like. So many proteins in our body are functional proteins. Those are in the tertiary structure format. Depend upon the layers and how they are folded. So we are assigning primary, secondary, tertiary like that. So now we will see how the amino acids are made up of. This is the broad view of how the proteins are made up of, and now how the amino acids are made up of. For example, I am taking an amino acid, so it's a common structure, central carbon atom, and you know in the organic chemistry we can see methane. You can see methane. What is the structure formula of methane? CH4. The central carbon atom is surrounded by four hydrogen molecules, hydrogen atoms. Now, these four hydrogen atoms are how they are replacing, and by what molecules they are replacing, we will see now. The central carbon atom, and this hydrogen will be same, and one hydrogen is replaced by carboxylic acid, COOH, and one hydrogen atom is replaced by ammonia, amine group, NH2, and this hydrogen will be R, variable. So this is the common structure of each and every amino acid, but we have 20 amino acids. We have 20 amino acids. So how they are varying? Because this is common, and this is common. And how 20 amino acids are different from each other, that will explained by this R group. For example, in this R group, A is there, so that is one amino acid. A plus B plus C is there, that is one amino acid. Depend upon the functional group which is attached to in the place of R, so that amino acid is different from the another amino acids. We will see the example of different amino acids. For example, we can see an amino acid, glycine. So common structure, NH2C, COOH, NH, and here in the place of R, this is a very simplest amino acid. So in the place of R, we have hydrogen. So by seeing H, remaining everything is same. By seeing H, we can decide that as a glycine. And take another amino acid, that is alanine. Same structure. This is everything is same, and only the functional group in the place of this R, here we are having CH3, methyl group. If someone asks the functional group is methyl, which amino acid it is, so you have to tell that is alanine. And another amino acid, serine. In this R group, that is replaced by CH2OH. So if the functional group is CH2OH, what is that amino acid? You have to tell that is serine. So every three functional groups are same, but only one functional group varies from one amino acid to another amino acid. So depend upon the structure, we are calling it as a different amino acid. So this is how the amino acids are presented. Totally, we have 20 amino acids. Already I discussed total 20 amino acids. 20 amino acids. Now, what are those? Out of 20, 10 are essential amino acids. 20 are essential amino acids. And 10 are Non-essential amino acids. Out of 20 amino acids, 10 are essential amino acids and 10 are non-essential amino acids. But now recent times this list is somewhat different, but we have to follow according to our textbook. 10 are essential and 10 are non-essential. But according to recent data, this one is went to the non-essential. And to the non-essential. So we have to follow as per our textbook until unless they have to modify. Okay, so what are these 10 essential amino acids? K, V, R, L, I, F, T, H, M, W. That is lysine, valine, arginine, leucine, isoleucine, phenylalanine, threonine, histidine, methionine, tryptophan. So these are the essential amino acids. And remaining tyrosine, cysteine, proline, glutamine, glutamic acid, aspartic acid, asparagine, glycine, alanine, serine. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. So these are 10 and these are 10. These are essential and those are non-essential. So what is essential and what is non-essential? That means these amino acids, essential amino acids, they cannot be synthesized in our food. We have to take it from external dietary source. That way we have to take some fish, vegetables, milk, next eggs. So we have to take these amino acids from the external diet, but these amino acids can be synthesized by our body. So these are non-essential because already it is there in our body. These are essential because our body is not synthesized these, so we have to take it from outside. So this is the difference between essential and non-essential amino acids. This list is very important because this will be present in our chemistry also. Chemistry in everyday life or some chapter it is there. So this list is important. I will give you acronym KVRLIFT HMW. KVR lift HMW. These are the single letter words of these amino acids. And we have to know, first of all, now we discussed about the basic structure of amino acids and the list of amino acids. Now we will see how each and every amino acid, how they will attach to each other in the process of making a complex protein. How they are helpful for maintaining or manufacturing of complex protein. I am drawing here amino acid 1. So carefully observe. This is COOH, H and H2, and this is some other any amino acid R group. end. This is another amino acids. See, COOH is always at this end only, and NH2 amine group always has to present in this end only. For example, these are the two amino acids. So how the linkage, how the cement will be established between these two? Because how it means, out of these two hydrogens, one hydrogen, for understanding purpose, I am writing these hydrogens, this one and this one, like this. And now, how the cement is established, how the bonding is established means, this H is combines with this OH. So H plus OH results into the outbreak of H2O. It will release outside. And now this bonding will be established like this. CO HN. So this is one amino acid. This is second amino acids. So this bond is known as peptide bond. This bond is known as peptide bond. Understanding? So now this OH and H will be removed as outside in the form of water. And this H I am writing here. So this bond is a peptide bond. Peptide bond. So here peptide bond, we have to know, is formed by the result of or by the release of one water molecule. So that's why the hydrogen peptide bond is established. So this is how the cement, this is one brick, this is one brick. And how the cement is established? By removing one water molecule and formation of one peptide bond. So this is the peptide bond. And again, if another amino acid will come, this is another amino acid. Now this one will go outside and formation of one peptide bond. So this is how the numerous number of amino acids will come and results into the formation of complex protein, and all these amino acids will fold it together and forms a ball-like structure that results into the tertiary structure. That is the functional protein. Most active protein in our body are tertiary structural proteins. And now we discussed the structure and how it is formed. And in an exam point of view, they may ask how many number of peptide bonds are formed during the formation of a protein containing some X number of protein amino acids. So I am writing a question here. What is that? How many peptide bonds? How many peptide bonds will be formed during the formation of a protein contains 100 amino acids? So how you can calculate? There is a simple formula here, no need to worry. A simple formula, n minus number of peptide bonds. They are asking number of peptide bonds is equal to n minus 1. Here the n is equal to number of amino acids. So here 100 minus 1, that is simply 99 peptide bonds will be formed, because for example this is an amino acid and this is an amino acid. So this is an amino acid like this. Now calculate the number of amino acids. 1, 2, 3, 4, 5. N is equal to 5. And calculate the peptide bonds. 1, 2, 3, 4. That is equal to n minus 1 only. That is 4 peptide bonds only. So this is how you can calculate the number of peptide bonds. Or else they may ask, if somewhat they have to increase the toughness of the question, they can ask how many water molecules will be liberated, will be formed, will be released during the formation of a protein which contains 100 amino acids. Because same formula, because one peptide bond, one water molecule. So same formula, again n minus 1. N is equal to number of amino acids. So this is how simply, without losing a mark, you can calculate the number of peptide bonds and the number of water molecules which are going to released outside during the formation of a peptide bond. So this is a protein biochemistry. Nothing is there in the protein biochemistry. We have to know the basic structure of an amino acid. So once you understand the basic structure, what is the difference between different amino acids, we have to know. That is simply that R group. In one amino acid, the R group may be simply H. In another, it is CH3. In another amino acid, that is CH2OH. So due to the variation of that R group only, we have different types of proteins. And any amino acids can fuse each other and results in the formation of peptide bond. Any amino acid can bind together. Understanding? So this is about protein chemistry. And now the basic concept why we are discussing about protein biochemistry after transcription. So slowly we are going into the topic of translation. Now that process, if we are going into the translation, we have to start it with genetic code. Genetic code. What is genetic code? And what is the use of a genetic code? And what is the significance of genetic code? So here we have to know that how the amino acids will be formed and how many codons are there in the amino acid and how the translation process we will see now. So before translation, so this is the translation process. So before translation from mRNA, so what is happening to come here? Because from DNA already we saw how the mRNA is formed, but from mRNA to translation, that is protein. So what is required? What is required? So for that purpose, we have to understand some depths of molecular biology. So mRNA, I am drawing an mRNA here. mRNA contains the nucleotide sequence only. So for example, U, A, G, C, A, C, G, A, U, 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 and A, G. So this is an mRNA. Briefly understand, but tomorrow we will, in the next class, we will see the detailed process of translation. Now, this is mRNA, that is from the nucleus, it is going into the cytoplasm. So after going into the cytoplasm, how the proteins are formed? How the proteins are formed? So this is mRNA that is present in the cytoplasm. Now what happened? So many simple nucleotides are present in the cytoplasm. So that nucleotides will be bring by tRNA, that is transfer RNA, that is bring by the tRNA, and all together will reach us to ribosome. All together will reach us to ribosome. Yesterday in the WhatsApp group I had sent an animation video. Ribosome is like this and this mRNA is traveling like this. And during the traveling, the tRNA comes and gives one and another codons. What are the codons? We will see now. So now what will happen? For example, I am taking UAG. This 3. Now tRNA will come and it also brings the complementarity to this. For example, complementarity U, A, U and C. So these 3 nucleotides it will bring and comes here and attach it to here. And that triplet, this is triplet, 3 nucleotides, that is triplet. That triplet is known as codon. So one codon is responsible for the formation of one amino acid. Understand? One codon is responsible for the formation of one amino acid. So how many codons are there? How many, like that, how many codons that tRNA will bring and enters into the ribosome and it will forms the protein? Now that thing we have to know now. So totally that number of codons, that is genetic code, totally we have 20 amino acids only. 20 amino acids only. Now, as we are discussing the formation of protein from mRNA, it is not directly from DNA. So we have the nucleotides U, uracil, cytosine, adenine, guanine. 4. And I told that one codon contains 3 nucleotides. 3 nucleotides. So these are 4. Basically we have 4. In codon we have 3. So 4 into 4 into 4. That is 64 codons we have. This is very important. Total 64 codons which are responsible for the formation of different amino acids. So once the different amino acids are formed, different proteins will be formed. So for the formation of amino acids, these codons are very important. So how they are formed? Carefully observe. See, this is a table 1, 2, 3, 4, 5, 6, 7, 8, like that 16. U, C, A, G. 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 Like this. Now, how each and every codon will be formed? U, U, and this U. U. Next U, U, and this C. Next U, and this U, and this A. 1, 2, this one. U, U, G. Now, 4 codons are formed. 4 into 16, 64. And another box. U, C, U. U, C, C. U, C, A. U, C, G. Like that, take any box. For example, this one. G, A, U. G, A, C. G, A, A. G, A, G. Like that, the codons will be formed. And from the codons, the amino acids will be formed. And from amino acids, we are having proteins. And what is the importance of these codons? What is the importance of this genetic code? How they will form, that we will see in the process of translation. But now we are discussing the importance of the codons. What is the specificity? And now here, for example, see here. We have 20 amino acids. One codon will form only one type of amino acid. See how much specificity is this. U. It will form only one amino acid, that is phenylalanine. And U cannot code for alanine, and U cannot code for valine. So this is the specificity. One codon, triplet codon, will be responsible for the formation of only one type of amino acid. See, one codon leads to formation of one amino acid. But here you have to observe carefully. One amino acid can be formed by so many number of codons. For example, this phenylalanine will be responsible by all these. So this can also form. This can also form. This can also form. Carefully, you have to observe. You have to understand the difference here. See here, UUU can are coding for phenylalanine. UUC, UUA, UUG, all are coding for phenylalanine. But that means this one is coding, this is coding, this is coding, this is coding. But UUU at the same time it is not coding alanine. This is not coding alanine. That means one triplet codon will be codes for only one amino acid. But one amino acid can be coded by so many number of codons. So that's you can observe here 64 codons are there total, but we are having 20 amino acids. See here, you can observe total 64 codons, but amino acids are only 20. So it means some other codons, some more number of codons are coding only more number of proteins. Understanding? So this is more. That means more number of codons are coding for a particular amino acid. So this is the difference you have to observe. So that is also the property of an amino acid. So we have some properties. We have total 64 codons. Out of 64 codons, 61 codes for amino acids, but other 3 codons are acting as a stop codons. Stop codons. Example, in the transcription process we have termination factor. Like this, once the protein structure is established, it has to check that process. It has to stop that process. So these 3 codons are helpful for to stop the process of translation. So what are those? UAA, UAG and UGA. These are the 3 codons which are helpful for stopping the process of protein formation. And remaining 61 will code for different types of amino acids. So this is one of the property, and this is the second property. That is, one codon will be responsible for the formation of one type of amino acid. So that is one codon property is degenerate. Degenerate. That means, for example, one codon will be responsible for the formation of only one type of amino acids, not any other type. So either it will code or it will not code. It will degenerate. So that is how one property is assigned for the specificity of the codon. And these codons are contiguous. That means here, for example, this is a sequence. Like this. The tRNA will come and the ribosome will read in a continuous manner. It will not stop unless until when this codon has to reach here. Unless until if the stop codon is not there, it is continuously has to read. So that's why the genetic code is contiguous. It has to read 333, 333, 333 like that. If it is not read like that, for example, due to some malfunction, it is reading these 4 together, then some other protein which is very harmful to our body will form. So it has to take the codon in a triplet model only. Because one amino acid will be formed by this triplet codon only, not by 4 nucleotides. One codon contains only triplet, not 4 sets. Okay, so this is how the sequence has to be read continuously. And another specificity of the genetic code is we have dual functions. One codon will be act to code an amino acid, and it is also acts as an initiator. So what is that codon? AUG. AUG is a codon that codes for an amino acid methionine, and it is also acting as an initiator codon. For example, each and every protein has to be formed from AUG only. If AUG is not there in the sequence, that process will stop at that time only. If AUG is there, then only it will continue the process further. So it is acting as an initiator codon, and it is also codes an amino acid methionine. It should be either here also, but at the starting place it should be there. Then only the ribosomes will allow the formation of protein synthesis. So this is about the importance of the genetic code. And one more thing, universal. The genetic code is universal. So what is the meaning of this? The genetic code is universal. This means whatever the animal you can take, from bacteria to human beings, and in plant kingdom, in fungal kingdom, whatever the organism you can take, that codon will codes for that amino acid only. For example, you take UUU and observe what this codon codes for in bacteria. In bacteria it codes for phenylalanine. And see, in human beings, this UUU codes for the same amino acid. We can take whatever the organism, but it is coding the same amino acid. So that's why the genetic code is universal, from bacteria to human beings. So these are the special characters that are present, and the specificity. See how much specificity. If I asked you to do work continuously for 1 hour, after 59 minutes or after 40 minutes, your mind will be disturbed and you will commit a mistake. But lifelong, with this much specificity, each and every cell is doing the work. See how great they are. But even they are not visible also, very microscopic, and they are doing the functions very specificity, but we are not doing that. So this is about the genetic code and the facts and the specifications of the genetic code. And in the next video we will see about the process of translation, how the tRNA will bring, how the ribosome plays an important role in the formation of proteins, and how the mRNA is transported from the nucleus to cytoplasm. So all these details we will see. Now by this video we are knowing that what is the protein biochemistry, how the proteins are made up of, that is amino acids, how the amino acids are made up of, and how the two amino acids are linked with each other, and how different types of proteins are there. That depends upon the structure of this amino acids bonding, that how many amino acids are present in the peptide chain. So based upon that, it is in a primary, it is in secondary, tertiary. If only one amino acid strand is there, that is a peptide chain, so that is primary, and only 2 or 3 layers, that is secondary. And if you ball like structure, that is tertiary. And in our biosystems, that in every organism, if the functional proteins are there, that will be in a tertiary structure. And that is about the protein biochemistry. And now how the proteins are formed? Amino acids. And how the amino acids will be formed? Due to these codons. So how these codons are formed, and how the tRNA will bring different nucleotides and add with these, which are helpful in the formation of amino acids, we will see in the next video. If you have any doubts, text me in our chat box. Thank you so much.